Good afternoon. I'm Alessandro, and today I will talk to you about a new EGG data analysis pipeline that I'm working on during my PhD in Rome. So, fortunately for us, you already know a little bit of EGG thanks to the previous presentation. So it is, as you know, it is a technique to record electrical activity with the help of surface electrodes. And as you already know, typically at rest and fasting, you have this cyclic activity of at around 0.05 hertz that is called normogastria. And this activity can be modulated and disrupted by a lot of different factors, including, for example, digestion, nausea, emotional state, or gastric disorders. And also, giving the things that we learned this morning may be of interest for you, also by the food intake, not only the type and the quantity of food intake, but it should also be modulated by the caloric content of the food intake. But I see that there are experts in the room, so maybe you can say better than me. So it could be of interest also in context of nutrition. But its responsiveness to a lot of different factors, both physiological and psychological, and together with EGG non-invasive nature, makes it a very powerful and technique with a lot of potential in both clinical and research applications, but it has some limitations. And I think one of the main limitations is that the EGG signals is notoriously difficult to analyze for many reasons. One of them is that the signal is generally very weak. Actually, is much weaker than the cardiac signal. For example, is on the scale of the microvolts. Also, it is a signal with ultra low frequency, so it requires you to record for a long period of time. And the main problem, I think, is that there are a lot of strong source of noise that can disturb the signals, and this source of noise have an overlapping frequency spectrum that overlaps with the EGG signals frequency band of interest. Some examples of them are, for example, general electronic interference or also electrode contact resistance with the skin of the participant, but also participant movements. And these interferences are very strong and cannot be ignored because they substantially undermine the result of the analysis. And so for this reason, although there have been a lot of proposal in the literature, there is, not yet a gold standard of analysis for single channel EGG data. And so my goal is at least to get closer to this gold standard with my work. By gold standard, I mean an analysis pipeline that is rigorous, data-driven, and it's as much as objective and reproducible as possible. So first of all, we have to recognize, to start the analysis, that the EGG signal is not stationary. And this means simply that its properties are varying with time. They are not constant. So time frequency analysis methods are needed. Here you can see, for example, a real recording EGG signal from our lab, but it's not until you do a proper time frequency analysis that you can understand basically what is going on. And on the plot on the right, you can see a very clear dark blue band. It represents a pretty steady dominant frequency of the stomach. On the right, you can also see the power spectrum curve, a more traditional study curve. And on the bottom, you can find another quantity that I introduced that is simply the energy of the stomach activity. I called it the instantaneous energy curve because it gives you the energy of the activity of the stomach for each instant of time. And this will be a very important quantity in the next slides. To get this, I used a technique called super electron spore that has been proposed recently because it has a much higher time and frequency resolution respect to traditional time frequency analysis techniques. And this is crucial for us because you need that precision in order to localize precisely the anomalies and to get rid of them. So let's focus on this curve. Here you can see simply a zoom-in plot of this instantaneous energy curve for the previous signal. That is a very clean one. On the other hand, here you can see another signal that is very clearly corrupted by artifacts. And you can see that the curve, and you can see also the instantaneous energy curve on the right. And by eyes, you can see that in the first case, this curve is pretty much uniform. But in the second case, it has a lot of peaks. And so we can, in order to understand if a signal is contaminated by artifact or not, we can look at these peaks, basically. And of course, you can get a quantitative measure of this. And I propose to use a quantity called Zell's entropy. That is a mathematical quantity that has the properties of being lower when the signal has a lot of peaks. So in this way, we can understand if the signal is clean or not. Once we know this, our goal is to localize the anomalies in order to get rid of them. So in order to do this, we can do simply a scan, let's say, of the signal with a moving window, like the one you see here in red. And we can simply compute the values of the signal in this time window. You can also produce an histogram, for example, and considering the point at the center, one that you can see there, that point has values that are very extreme. So this point can be flagged as an anomaly. On the other hand, moving on other parts of the signal, you can do the same procedure, and considering another point, in this case, the value of this point is not that extreme, so that you can flag this as a normal point. So doing this for each point of the signal, you can identify the intervals of time where anomalies are. So you can get rid of them. And so you can move from this situation in which you have a clearly contaminated signal with a lot of anomalies, with a very strange power spectrum, to this kind of signal where you remove the specific time points that are anomalous. And as you can see, you can recover a nice Normogastrian activity as signals in the, it is signals in the, look at the power spectrum, there is a peak in the typical gastric frequency. Once you have a clean time frequency representation, you can extract from it a lot of parameters. And here I simply represent some of that I propose, some explorative quantities with a spider plot. And you can find, of course, the traditional ones, for example, the dominant frequency, that is I think the most well-known parameters in EEG analysis. It represents simply the frequency at which you have the maximum in the power spectrum. So it's the main frequency component of the signal, let's say so. But you can also consider some other traditional parameters like the dominant power. That is simply the value of the power spectrum at the dominant frequency. But you can do more. For example, you can compute another quantity called Renyi entropy that is very used in the time frequency analysis contexts. And it is simply a measure of concentration of the time frequency plot. So, for example, just to give you some hint about the quantity, and the signal on the top, it is very low entropy because the signal is very concentrated along the time and frequency. It has a very steady frequency. The signal on the bottom, on the other hand, is very scattered. This energy is very scattered along the time and frequency dimension, that it has a very high entropy. So the point of calculating all these parameters is that in this way, we can sort of define an EEG fingerprint for a singular EEG channel, and we can characterize more fully the dynamic of the channel. By the way, in the light of the previous presentation, I didn't have time to explain it, but to put a plot. But we can also compute the instantaneous quantities, like, for example, the instantaneous amplitude, phase, and we can do it directly from the time frequency representation, for example. So it could be useful. Yeah, so it's the point is that to fully characterize with more sophisticated methods the dynamics of the EEG, and so to get a lot more information, both for psychological and clinical research. So yeah, I think that's it. So thank you for your attention, and a special thanks also to my collaborators.
Okay, questions for Ale?
Like a fantastic pipeline. Have you guys thought about integrating it in addition to the BEP rhythm with other types of rhythm? And how would you the alignment between different rhythms?
Do you mean rhythm for the from the
Like,
body?
say, for example, heart rhythms or breathing rhythms.
Yeah, I mean, this of course you can analyze this pipeline independently from other body signals, but yeah, the goal, for example, in the future, also in my lab, is to do a phase amplitude coupling with the EEG signals. But yeah, it will be very interesting to me also to couple it with other body signals, for example, respiration or heart signals. And it is usually not done. And I think one of the reasons, for example, is that they have very different time scales, for example. And you, for example, in heart rate variability analysis, you don't have a continuous measure of the instantaneous the frequency of the signal of each instant of time. So you cannot correlate it. In EEG, you have a lot more time resolution. So yeah, I'm thinking about doing it in the future, but this will mean that it is needed also to analyze the other body signal with more time precision, for example. So it requires more work, of course, but yeah, it could be a very interesting direction to study.
You mentioned that
Yes.
with an understanding of the time resolution of tendon rhythms and their relation to particular solutions.
I don't understand if I get the.
So if you have different time courses,
Yeah.
this relates to potentials and frequency
Mhm.
changes. So heart is faster than the gut,
Yeah.
and the brain is even faster than the heart. But in the brain EEG domain, we were talking a lot about frequency coupling,
Mhm.
and they're obviously also different.
Yes.
So is that a way forward? I'm trying to prompt all of these.
Yeah, I mean, the point is.
This would be a way forward.
Yes, yes, because the point of this phase amplitude coupling is precisely that you can get coupling with different scales. For example, he talks about alpha band, but you can also find some other couplings. So the
Slower.
yeah. That's the point of the frequent the phase amplitude coupling is just one of the possible couplings that you can compute. But the point is, the interesting part is exactly the fact that you can coupling with the phenomena that have a very different time scales. So that's the point of getting this type of analysis. So yeah, in the future, I would like to study more this.
Yeah, I have two. So like partially as a follow-up of what was
Mhm.
discussed now. I mean, in a sense, in the EGG, as we also saw from the slide before, no, from these papers from Rebollo, Azzalini, and so on, they always show this figure where from the EGG, more or less reliably, you can get surely the ECG, the gastric signal, but also a respiratory signal. And then, you know, now all the cardiac activity, and usually the cardiac activity is used to simply remove artifacts from the EGG, but in a sense, it's a signal that is there and could also be used for quantifying this coupling, maybe with the same sort of time and frequency resolution.
You mean that you can obtain for example, the heart signal from this? Yeah, you can.
As well as the
respiration. Yeah, but yeah, I think that if you have more precise technique that are dedicated to them, yeah, you can extract information because here it is a very clean signal, so you can clearly see everything. So it's not always like that, unfortunately. But yeah, here you can see clearly the hard peaks. So the very, but I don't think there is a particular advantage to extract the hard signal from this recording and not from, for example, a standard ECG, or I mean, for respiration belt or whatever. There are a lot of techniques. If it is the question, I don't know if.
No, in a sense, I mean, if you have the
But you
one
can.
recording and then you want to use the same method
You can.
from the same.
You can, because here I restrict the time frequency representation.
Yeah.
Maybe you can't see because they are very small, but from only in the frequency band of interest, it's like 0.03 to 0.16. But yeah, if you enlarge the representation, you will also get the cardiac and respiratory component. Yeah, so you can principle.
The other question is, I mean, I like how you started. That is the fact that we use methods for for stationary signals, with all the assumption of stationary signals like FFTs, and then you propose the alternative. But then at the end, what we end up with is again like a 0.04 hertz. There is a peak in the spectrum, and it's also usually what is used in EEG. So I mean,
yeah,
the whole story about stationary and station. No, what I want to ask is the other metric that you're proposing, instantaneous energy, is it thought to reflect something else? That is more the motility, is about digestion, is about something else in addition to the frequency? Because if we only look again at the peak frequency with the entire history, neither fluctuation around.
Yeah. Now, first of all, for the motility measures, for example, there are some proposal about a new technique called high density gastric recording, for example, in which you can also compute the velocity of propagation of those low waves for the stomach. You cannot do this in a traditional EEG analysis, but yeah, the quantities that I propose are not limited to the dominant frequency. There are a lot of different characterization. For example, here in the normogastria brain, you have a very, let's say, simple power spectrum that can be characterized with a lot of precision by the dominant frequency. But in other contexts, in which you don't have a clear peak, for example, you need a more fully characterization of the curve. And for example, there are some entropy measure of the curve that can be very useful. Or for example, this just anticipate that, for example, the thing that I showed before, the Rayleigh entropy, that is a measure of concentration at the frequency plot. I'm using it with our data, and it seems to be very sensible to a sort of emotional activation that I don't can speak about the details, but of course, right now they are all exploratory quantities, and sorry, because they are not still in the literature. So we don't know exactly at the moment what is the meaning to assign to them, and they are not maybe such direct as, for example, dominant frequency. But yeah, but I think that using it, you can discover some associations that are not present and not known today, for example.